I will start today's lecture that is focused on databases. We will focus on chemical databases, and we will talk also about quality of 3D protein structure in the databases. Generally, the chemical information systems are governed by chemoinformatics, which is the application of computational tools to deal with chemical data, with the connected biological data to structures, and also designing library or performing virtual screening. All these processes require effective storage of structures and data, precise retrieval of structures based on specific search criteria during searching the databases. And the chemical databases also enable calculation of structure-derived data and dealing with these data, searching these data. An additional property of some databases is also execution of in silico chemistry operation and enumeration of combinatorial libraries. How the databases can look like. In the first column of this table, we have the operations. So firstly, we deposit, we store the data in the database. Then we retrieve them and search, and we can also deal with the relationships. So how the classical information system work, it is visible from the second and third column. So you only deposit words or some numbers, for example, dates, and you are able then to retrieve based on the number of record or entry, based on some substructure search or find a specific answer connected to a specific term. In the two right columns, it is visible how the chemical information system works. So we store the structure of a chemical compound. It can be stored in the SMILES code or any alphanumeric line entry. There are also other codes of this type, and you can retrieve then back the structure of a specific molecule. You are able also to perform some substructure search, which is in the third line, and you are also able to work with the data or parameters calculated in the database. This is a figure that I copied somewhere in an article, and you can see that you can deposit different data for chemical structures. It's the structural data, in vitro biological assay results, in vivo results, some physicochemical properties or any other parameters. The database needs to have a strong searching engine, and all the data should be supported by references. So I suppose that this is already known for you, for example, from the basic course of pharmaceutical chemistry, how the Reaxys database or Cyfander database works. Let's talk now about the methods of structure determination. So for solid states, small molecules, the most suitable methods are X-ray crystallography or neutron diffraction. For gas phase molecules, it's microwave spectroscopy or electron diffraction. And for proteins, the most used method is X-ray crystallography, NMR, and cryo-electron microscopy. For small molecules or generally any molecules, the positions of particular atoms in the 3D structure are based on XYZ coordinates in the space. So we use so-called Cartesian coordinates, which is the position in X, Y, Z space, defined space. For small molecules, we can also generate the 3D data. Apart from X-ray crystallography, we can use some software, for example, for conversion of 2D data of molecules into 3D shape with using some automatic approaches, or we can use fragment libraries to create the geometries of the whole molecule. So let's talk about the most used method, which is X-ray crystallography. All the terms that are in blue in the presentation are interactive links to Wikipedia, so you are able to read more about the method if you are interested in. And the principle of X-ray crystallography is scattering of X-rays by the electron cloud around an atom. So we use X-ray beam to irradiate a well-prepared protein, and we get a structural pattern. By this method, only heavy atoms are visible. So it means that the hydrogens are not visible as they are too small for this method. If in case we work with the atomic model of molecule resolved by X-ray crystallography, we always have to check if all atom types are correct, if the bond lengths and bond angles are reasonable, and if bond orders are correct. We should also check the chirality of the molecule and so on. So from this method, we can get a molecular data file that contains the position of all atoms in the molecule and also some details about the experiment and also about the organization and so on. So this on the left side, you can see how the output from this method can look like. This is the X-ray diffraction pattern that is elaborated by computers and specific software, and it gives then the atomic model of the molecule. So the X-ray specialists get from this experiment the idea where the electrons, electron clouds, and the occupancy of the particular atom, and based on the knowledge of amino acid structures, they fit in the amino acid backbone and the residues. And they have to check everything and maybe also correct. NMR spectroscopy is suitable for small or medium proteins because with large proteins it's too complicated to resolve all the peaks in the spectrum. And the advantage of this method is the fact that it provides information on proteins in solution. So you do not need to prepare a purified crystal, as in case of X-ray crystallography. Okay, I have a comparison of two outputs. The first one is from X-ray crystallography, the upper one, and the lower one is from NMR experiment. So it's obvious that no hydrogens, yeah. However, the softwares working with these molecules are able to add where they should be, where they fit. We talked already about the 3D cryo-electron microscopy in the first lecture. So maybe you should know the 3DEM abbreviation, which means 3D electron microscopy. And we already mentioned that this method requires preparing of thin layer with protein in the solution in a non-crystalline ice, and it affords the overall shape of the protein as it is visible in the figure. So the experts are then able to observe the crystal using the electron microscope. Okay, we mentioned already a little bit the conversion of 2D data into 3D using computers. So we can use different softwares. It is sufficient to have only the SMILES code that can be transformed into 2D structures and then transformed to 3D. Even the package of Chemofist, whose part is ChemDraw and 3D Viewer, is able to convert the 2D structure into 3D. The software we use here at the faculty, MOE, is also available, and there are plenty of these softwares. As for the formats, some general formats for the storage of structures is, for example, MoFile, molecular file format, or SD format, which is structural data format. The PDF format is used in the PDB database. We will be talking about it today. I mentioned also creation of 3D structures of small molecules based on fragment libraries. So we can construct the 3D shape of small molecules using some known geometries of of very small parts of molecules. Here is an example of an acetylpiperidine, and you can see the crystal structure in the first figure, and you can see then different softwares, how the conformations were created. So I would say the last one is maybe the most reliable with the side chain. We can use the X-ray, NMR, cryo-electron microscopy for protein 3D determination, 3D structure determination. However, if we are not able to get either the crystal or to measure it in NMR in solution or to measure it in cryo-electron microscopy, we have some theoretical or like calculation engines how to predict the 3D structure. We call these proteins computed structure models. This is a relatively new term, so please remember the CSM abbreviation, computed structure models. And generally, we can use two methods. The first one is template-based modeling. We can mention the synonymous homology modeling. However, in the last few years, new template-free modeling methods have appeared. So, as for the homology modeling, it is a template-based method. Using comparative modeling. So if we, in case we know the amino acid sequence in a protein, but we do not know the 3D structure and we want to know it, we can find related homologous protein where the 3D structure is known. And based on the comparison, we can predict the 3D structure of the unknown protein. The sequence identity required should be at least 30% or 40% is better, at least. And in the table, you can see the dependence of the sequence identity in two proteins. One is known, the 3D is known, and one is not unknown. How it is reflected in the RMSD, which is the root mean square deviation of the protein. So the lower RMSD, the better. So with knowing the 70% of the sequence, we can have a quite reliable model with low RMSD value, whereas with knowing only the 25% sequence identity, the model will not be as reliable and the RMSD will be higher. How does the workflow for homology modeling go? So firstly, we have to find a template, at least one, but we can have also more templates to get a better and more reliable model. So you can see it in the figure on the right side. we have a template where the 3D structure is determined, and we have a protein where we know only the amino acid sequence, and we want to predict the 3D shape. So the first or the second step is the alignment of the amino acid sequences, or which is also visible in the figure. And based on the amino acids alignment, we are able to construct the 3D model. So firstly, we generate 3D coordinates based on the model. And in case there are some sequences that are not in common in both the proteins, we have to use the ab initio modeling for the loops, turns, or other areas where the model cannot be supported by the template.
By which method we can know the protein sequences before we get to that stage?
Sequencing.
Sequencing is another method that can be used.
It's biochemistry method. So actually, the genome of a man has been already sequenced. So we know the amino acid sequence for all the proteins in the human body. As for bacteria, for example, it is under process. This is the job of biochemists.
Yeah, we have library.
Yes, we have libraries for this. You can see that the computed structure model on the right bottom side of the slide was created based on a comparison with the template, and there is an extra loop that was predicted using ab initio modeling. What is required at the end? It's the model quality assessment. Some softwares, for example, MOE, is able to create some small loops up to specific size of the oligopeptide. However, there are different web pages for surveys that will do the job for you. For example, Swiss Model or FIRE2. Our colleague is using the Swiss Model website for homology modeling.
The template should be from the crystallography X-ray, for example, or
yes, experimentally determined. We use as template experimentally determined structure.
For example, in the unknown structure, when we know that it could be a template for another stuff.
So it depends on the sequence identity or similarity, and also on the function of the protein. The protein should be similar in the function.
Okay, I'm thinking about that after many generations, after many processes. For example, it's not something like verified after that with that sequence, which is we don't know it's 100%. It's a possibility.
It's prediction.
It's prediction.
It's prediction.
After many generations, after many prediction, the thing that became a template for another one, which is more far from that.
It happens for some proteins that, for example, now the 3D structure is not known, and only homology modeling can be used for the prediction, and maybe in 2 or 5 years the protein will be determined, the structure will be determined using some experimental methods. So we can then reflect, which already happened in the past, after the experimental measurements, we can then reflect how the homology model was reliable. It happens. Actually, I forgot to mention that the template is from insect. It's a protein from insect, and the right one was human. human protein. So it was again example from an article or textbook. Okay, this slide is a little bit talkative. However, in the last few years, new methods have been employed in protein modeling, and they do not use any templates, but they use, for example, artificial intelligence, which are the neuronal networks, or some deep learning, machine learning. So, and the methods are based on some learning from known proteins. It's usually connected to the PDB database, and using the neuronal network for sequence alignments of small parts of the protein, we can predict the 3D structure. So one of the softwares is the Rosetta Fold using deep learning. And this year, the AlphaFold was published, which is artificial intelligence system developed by DeepMind, which is the daughter company of Google. And they claim that they are able to predict 3D structure of any protein. So I'm not sure about the maximum size, but you would find it probably in the website of AlphaFold. And they use in an additional refinement process, they use the combination of artificial intelligence and machine learning. A complicated topic. I do not like to go much into detail. Okay, so we mentioned the experimental methods for structure determination, and we mentioned also some theoretical methods for structure prediction. This is an overview on the left side, how the process goes in experimental methods, and on the right side, it's for the calculations. The IT specialist focused on the artificial intelligence, machine learning, deep learning can involve not only protein data bank data, but also the genomic data, which is the connection between proteins and genes. And they publish the predicted structures either in articles, but they are also available in databases. I will show you them later. Okay, so let's start with some databases, so some specific databases. The first one is the Cambridge Structural Crystallographic Database. It is released by Cambridge Crystallographic Data Center, and it is the most important database for crystallographic information of small molecules. So you would find here, for example, the 3D structures of drugs or any small molecules. It started 50 years ago in Cambridge by Olga Kennard, and you can go to the website from the link and also to some educational sources. I would like to show you some statistics from this database. So at the moment, or in the year 2020, there were more than 1 million of entries. This number, 1 million and more than 200,000 is the number from this year. So it involves entries of experimentally derived coordinates for organic and inorganic compounds up to the size 500 atoms. So low molecular compounds, so-called small molecules. And every deposited entry contains bibliographic data, connectivity data, molecular geometry data, and it's downloadable in safe format, which is crystallographic information format. So this is how the database look like, and I think we can open the database now. So this is the website, and we can access structures, and we can type, for example, paracetamol compound name, and the database retrieves several entries. For example, different salts. We can also find here only the molecule of paracetamol. If you click on the entry that you are interested in, you are able to observe here the 3D structure, and you should be able to download also the crystallographic information file when you put the email. So you are not able to download more molecules at once, but only one entry for free. So I would like to continue now with the Zinc database that was originally designed for virtual screening. And it is a very important database for us because it involves over 230 million purchasable compounds in ready-to-dock format. So we can get set of molecules in the 3D format, which is not, I think, possible from any other database. This database is provided for free by the University of California, San Francisco. And you can also search the database based on specific subsets. I'll show it. So we can choose, for example, compounds that were tested in cells or that were tested in human or in vivo generally. We can download a set of natural compounds, endogenous molecules or metabolites, or we can download compounds that are immediately available in the market or that are available on demand. And we can also use so-called branches, which enables filtration of compounds based on log-P and molecular weight. So this is how the website looks like, and where you will find the substances and branches. And I would like to go to the website again. Okay, so this toolbar might be interesting for us. So I click substances, and when I click subsets, so here you can filtrate from the whole amount of the compounds contained based on availability, bioactivity. You can, for example, filtrate FDA-approved drugs or world not FDA-approved drugs, investigational compounds in trials, and from different bioassays. There are some other criteria, for example, the reactivity criterion, and so on. So as for the availability, we can download only compounds that are immediately purchasable, compounds that would be prepared on demand. This is the so-called boutique subset, and so on. And I would like to show also the franchise, which is the table with where you can see the dependence of log P of the compounds on molecular weights. So you can choose different subsets. For example, the compounds here, I would get the compounds up to log P of 5 and up to molecular weight 500. And in the upper offer, I can choose the 3D format, either the 2D or the 3D format. And the compounds are downloadable in CSV file. So I would like to continue with another database that is very important for us. It's the Campbell database. It's provided by European Bioinformatic Institute, which is part of European Molecular Biology Laboratory. It's an European initiative, and this database offers open data of binding and functional and bioactivity data. In general, there are 2.3 million of compounds, but you can search the compounds also based on the type of assay, based on the target, and indication, and also some other criteria. So this is how the website looks like, and I skip to the internet. So clicking on the bubbles, you would get directly to the compounds in a table arrangement. And you should be able to download the CSV file from this website. And it is very, very interactive, because on the left side, in the left-hand column, you can filtrate from the whole amount of the compounds based, for example, on Lipinski rule of 5 violation, molecular weight, log P, and so on. So this is Campbell database. I would like to mention now some other databases. However, the already mentioned are the most important ones for small molecules. So as for the drugs, you can use a Canadian database called Drug Bank that contains 14,000, more than 14,000 drug entries, and also some biologics, nutraceuticals, and some experimental drugs. So you would be able to find the information on drugs freely available in this database. Here is the statistics overview focused on the particular type of compounds, and also the targets. This is how the database looks like. There are also different search criteria. You can either type the name of the drug or any pathway that is influenced by the drug, or any drug categories, drug classification, and so on. Enamine is a Ukrainian database that offers different compounds collections. The one is screening hits. It's intended for a real high throughput screen. So they are able to submit the compounds in vials for any screening. They also offer fragment compounds. You know that the other low molecular weight compounds up to 150 or to 200. And they have a collection of real compounds. They are actually not real, but the real name is derived from readily accessible through validated parallel synthesis. So this is an enumerated database, but the compounds should be synthesizable. It contains 31 billion of molecules. This company also offers building blocks, so you can order the chemicals for running the reactions, and some other services. You can check it in the website. GDB database. GDB database stands for Generated Database. It was started by Professor Raymond and his research group at University of Bern in Switzerland, and there were several releases. The latest, I think, is the GDB 17, and this database is enumerated, combining the biogenic atoms or elements that are usually present in drugs. And the maximum of atoms in one molecule is 17 atoms, and by different combinations they got 166 billion of molecules. So this is an approach how to firstly prepare the compounds in silico based on some criteria. So you would be able also to create larger molecules or smaller molecules, which was the case of GDB 11 or GDB 13. And use the chemical space to create new molecules. The question is if the molecules are really synthesizable. ChemSpider is another database of chemical structures. real structures provided by Royal Society of Chemistry. So I suppose that you would find there also drugs, but other organic compounds and their properties. The website looks quite simple, so you type and you will get some data for your molecule. PubChem is a database of compounds or chemicals provided by PubMed. It is governed by the National Institute of Health in United States, and you can find there small molecules, but also larger molecules, for example, nucleotides, carbohydrates, lipids, peptides, and other macromolecules. As the PubMed is freely available, this database is also available, and you can see some statistics at the bottom of the slide. And here are some other databases for small molecules. For example, the National Cancer Institute in the US also publishes the compounds that have some anti-cancer or anti-proliferative activity, and there are some other databases. So we, until now, we have talked about the databases containing mostly the small molecule data. I would like to turn your attention now to the Brookhaven Protein Data Bank that is operated by the Research Laboratory for Structural Bioinformatics. And it's the only worldwide repository for 3D structures of macromolecules. In the past, there were different, like a European part and Japanese part, but it was united at the website rcsb.org. And each structure is represented there by a 4-character alphanumeric identifier, like the PDB code. And the structure is downloadable in the PDB file format. So similarly to small molecules, this file, similar to some SD files or MOL file, this file also contains the Cartesian coordinates for every atom in the protein, also the connectivity data, bibliographic data, and so on. Last year, the Protein Data Bank celebrated 50 years. So, nice age. And it started in the year 1971 at Brookhaven National Laboratory. You can see that in the first 30 years, there were few entries in the data bank, and then in the mid of 90s, the growth started to be exponential. Nowadays, we have only linear increase in the entries deposited every year. In this year, there are 196 structures, more than. We can. I would like to also show the increase in the structures that were resolved using the 3D cryo-electron microscopic. It's really a very well-established method and most used, and the growth of the structures using the method for the resolution of the structure is really increasing. It's very popular. You can find different PDB statistics in the website. So I selected only the overall number of entries, the 3 electron microscopy entries, and here is an overview of source source organism of the macromolecules. So mostly the macromolecules come from man, from Homo sapiens. The second mostly represented are from cattle and so on and so on. This is how the web page could look like. So this is the 4-character alphanumeric code, so called PDB code. So this is a protein from an example of protein from Mycobacterium tuberculosis in complex with NADH. And you can see some data concerning the quality of the structure of the atomic model. You can see also some 3D rendering. If you would download and open the PDB file, so you would be able to see plenty of pages, maybe tens of pages, hundreds of pages, where every atom has its line with the number of the atom, with the element of the atom. You are able to see the XYZ coordinates, and you are able to see the occupancy factor and B factor, so-called temperature factor. So we will talk about it. You are also able to see which amino acid which amino acid the atom belongs to, and also the chain in the protein. So the first atom is the carbon with number 19 belonging to leucine in the chain A. And it's the 5th amino acid, so the amino acids are also numbered. When I skip back, you can see here the data. First is the method that was used for the structure determination is the X-ray diffraction. And we have some values. The 3 values below is the resolution, R value free, and R value work. So let's talk now about these numbers. So as for the resolution, when we evaluate protein structure in PDB, it's best to find the structure with the lowest resolution, which means that the structure is really certain. There are some other values that we can evaluate. It's the R value and R3 value, and you have some numbers, and you should remember these numbers, how much the value should be under optimal conditions. What can we also evaluate? The Ramachandran plot outliers. So when I skip back, you can see here a plot, and in the 3rd line, the WWPDB validation, The third line are Ramachandran outliers, which is here 0.4. This is not bad. But we can also evaluate side chain outliers and some others. However, the Ramachandran outliers are for us important, should be less than 1% of the amino acid. So let's elucidate the resolution. In the figure, you can see one amino acid, it's tyrosine, under different resolutions in different proteins. So at resolution 1 angstrom, the position of all heavy atoms is really certain. So the clouds of electron density are really tight, and the position of the atoms is more sure or more certain. Under the resolution 2 angstroms, you can see that the positions, the measured positions are more loose. With the resolution 3, the positions are even less reliable. So the lower resolution, the better, and optimum for docking is around or up to 2.5 angstroms. However, when you are searching the protein in PDB and you are not able to get better resolution than 3 angstroms, you will take the structure for your calculation. But when you can choose, it's better choose the structures with lower, which means better resolution. Okay, so I would like to explain now the R values, which is not so easy, but I will try. The X-ray expert measure the diffraction pattern, then elaborate it using a software, and based on the atomic structure they create from the diffraction pattern, they calculate back diffraction pattern for the model or for the atomic structure they created. And then they compare the measured diffraction pattern and the calculated diffraction pattern. And this is the difference between these two patterns is the R value. So under optimal conditions, the R value should be 0, no difference. However, this is too idealistic. There are always some artificial modifications in the structure. So the R value should be up to 0.2. Please remember this value. Yeah. And the R3 value, it's very similar. However, the X-ray expert excludes before the artificial modification, before the software elaboration of the diffraction pattern, 10% of the data, create the model, the atomic structure, and then they predict the diffraction pattern for the whole molecule and observe how the 10% are predicted, how well. So this value should not be higher than 0.26. So logically, it is higher than the R value. but should not be higher than 0.26.
But finally, if we have the information about the whole, why we much care about that 10% and we rely on that how
You have to get somehow from the diffraction pattern the structure. So some computational elaboration is required.
at the end the work will be finished, yeah, about the diffraction pattern of the whole thing, yeah. Okay, but you said that when 10% of the information which is available regarding this, according to depend on that 10%.
So
they go for all.
They leave out firstly the 10% before the artificial modification, before the computational elaboration.
Okay, let's see that.
If you remember the table with the Cartesian coordinates for the protein with the lines for the particular atoms, there was occupancy value, which says that under the condition it's 1, it means that the atom, the specific atom, is in the same position in every molecule of the crystal. So when the crystal is irradiated by X-ray, the X-ray expert is then able to see several molecules from the diffraction pattern, and they compare the structure of the molecules in the crystal overall, and they are able, if the occupancy value or occupancy factor is 0.5, it means that you can see it in the figure for glutamine or for tyrosine in the right figure, that it might be orientated either to left or right, and in case the occupancy value for this amino acid is 0.5, in 50% of the molecules of the crystal is the tyrosine orientated to the left, and in 30% to the right.
In case if you have more than one crystal form of that compound.
It's only from one crystal, but there are several molecules of the protein packed in the crystal. There is not only one molecule.
No.
So they compare, using the software, more molecules in one crystal. And when we work with some files from PDB, we should decide about the occupancy. We can choose the occupancy as users in the software. Okay, last value in the record of the PDB file was, maybe last but one, was B value or B factor, which is temperature factor for every atom, and it reflects the movement of the atoms. So when we compare the particular molecules in the crystal again, we can see that some parts of the protein are more rigid, and some of them are more fluctuating. So if the B factor or B value is around 50 to 20 or even lower than 10, the model of the particular atom is very sharp. However, it is when the value is higher than 50, it means that the atom is barely seen because it's in every protein molecule in different position. Maybe it could be understandable from this figure where the regions with in blue color have a low temperature factor, which means they are relatively rigid and the position of the particular atoms is reliable. However, on the surface of the protein, there are more flexible regions, some more flexible sequences, and they are colored in red or pink. So it's the temperature factor. So when I revise the criteria, how we can evaluate the protein, we mentioned the overall protein, we mentioned the resolution, R value, R3 value, and for the amino acid side chains, we mentioned the occupancy value, and for particular atoms, we mentioned the temperature factor. As I show you in the PDB, we have also the data about Ramachandran outliers. I already showed Ramachandran diagram in any other previous lectures. However, when I revise a little bit, in the protein backbone, we have the peptide bond. It's here or here, and we mentioned that the angle in the peptide bond is planar due to the electron shift. Because there is a negative, partial negative charge on the oxygen, which causes the shift of a low electron pair from nitrogen to the CN bond, which creates the pseudo-isomerism. And we already mentioned that the trans pseudo configuration is the favored one. The cis is very rarely, for example, for proline. And there are other two angles, dihedral angles, and it's the C angle, this sign. In English, it could be pronounced psi, which relates to the rotation of bond C and C alpha. So it's the carbonyl carbon and alpha carbon. And there is another dihedral angle that can be measured, and it's the phi angle or phi that relates the rotation of the bond from alpha carbon to nitrogen. So I would like to run now a very illustrative movie, 2 minutes. So I hope that everyone will be able to see it.
First thing to do in the demo is to orient yourself with respect to the alanine tripeptide model. This is your N-terminal nitrogen, and this is the C-terminal carboxylate group. And what we're going to do with this is we're going to do 2 successive rotations to show how atoms are going to bump into each other when we do these rotations. We're going to rotate phi, which is the angle between this central carbon atom and a nitrogen. We're going to rotate that from 180 degrees to 0 degrees. And then we're going to rotate psi, which is this angle from the alpha carbon to the carbonyl carbon by 180 also. Right now, all the bonds phi and psi are 180 degrees. So to do that rotation, the first thing we do is we can turn the model with the C terminal facing us and find phi, which is right here. And then we're going to make a twist phi by 180 degrees. So now phi is 0 degrees. And if we do that, we see that these two oxygen atoms now are very close to each other. They're bumping into each other. They're much closer than two non-bonded oxygen should be. So this angle, phi equals 0, psi equals 180 degrees, is forbidden in the Ramachandran diagram. Now let's try doing the same with psi. So to rotate psi by 180, psi is this angle, and we're going to change it from 180 to 0. Now we bring that around, and now we see that this oxygen atom in red and the hydrogen atom in white are very, very close to each other, and they're bumping into each other. So now we know that the angle 0, 0 is also forbidden in the Ramachandran diagram.
So from this movie, we can derive that there are some favored angles of C and phi angles, and it never happens that it would be 0. Yeah. So in the Ramachandran plot, we can see the dependence of phi, a value of C angle depending on phi, on the value of phi angle, and all the white areas are disallowed regions from the steric hindrance reasons. The yellow ones are somehow allowed, and the red ones are energetically favorable. So based on the atomic structure from any experimental methods, the software is able to calculate the Ramachandran outliers. It means the percentage of amino acids they do not comply with the Ramachandran plot. So the lower number of outliers, the better. And you can see that there are some allowed or favored value angle combinations for beta sheet, for left-handed alpha helix or right-handed alpha helix. So it's obvious that for a beta sheet, the value is around 180 because it's the zigzag, or like the straightforward going structure. However, in the helix, it's the structure always a little bit shifted to create the shape. And Ramachandran plot for glycine and proline looks a little bit different because glycine has no substitution at alpha carbon, so there cannot be any steric hindrance. And for proline, the proline nitrogen is involved in the peptide bond, and it is usually occurred or present in some turns or loops where the zero P value or zero C value is allowed. Okay, I would like to go to PDB, and maybe show you the surface of the website. So you can search any terms here. You can also open the advanced search dialog, which is more complicated, but sometimes useful. I mentioned that there are nearly 200,000 of structures. If there is the flask sign, it means they are experimentally determined. However, you can access in PDB also the computed structure models from either homology modeling or from the AlphaFold using the artificial intelligence. If you probably switch on this button, you will search also the predicted macromolecules. So I suppose the default mode is to search only the experimentally determined structures.
What's the difference between predicted and artificial?
Predicted, it's the predicted structures are elaborated using either homology modeling or the artificial intelligence, machine learning, and deep learning, the AlphaFold software that I mentioned. So there is a icon with the computer screen for the predicted structures. So I can type, for example, cyclooxygenase or anything, any enzyme that you would like to hear about. And I can choose any. On the left side, I can filtrate if I want to get the human protein or any animal's protein, or which method was used for the structure determination. I can also filtrate the resolution, which is very useful. However, I will take one of the entries just to show you how the website looks like. So very important for identification is the 4-character alphanumeric code, which is unique code for every macromolecule in the PDB. In the title, you are able to see what is involved in the structure. Sometimes it's it's only the enzymes, sometimes it's a complex with any drugs or any ligands bound into it. You can see from which organisms it comes, maybe also the expression system, what type of enzyme is it, like the classification. And you are able here to evaluate the resolution, R3 and R-work value. And in this tool, you are able to see the number of Ramachandran outliers, which is below 1%. So it's quite well. This is article where the structure was described. So the structures, experimental structure, determined structures are firstly published by the researchers in any journal. It's usually some crystallographic journal. In this case, it's biochemistry journal. So you should be able to find the original literature and to get information about the overall research, what the researchers performed within the work. You can see how many chains in the protein. So we have A and B chain here. Length of the sequence, We can see here also some additives that are used that are either like residues in the protein from the organism or some compounds that are used for to support the crystallization. And you are able also to see all the small molecules involved in the protein. So in this case, I think the drug is probably the sulfonamide. And here are some more details about the experiment, about the X-ray experiment. Which you also find in the toolbar: experiment, sequence, and so on. So okay, it's not so much interesting. However, here you can find the second toolbar. You are able to find a viewer where you can rotate a little bit and find the small enlarge and to find the ligands. For example, here is the hem involved in the structure, and we would be able also to find here the sulfonamide. And you can change the rendering mode, like the type of the model in the structure and others. And you should be able to download the whole complex as PDB format, which we will do in the seminar. So I hope that I did not forget anything what I wanted to deliver you. I excerpted data from different articles, textbooks, but also from education web page of PDB. It is the PDB 101. So here are some short movies focused on the 3 experimental methods like X-ray NMR and 3D EM.